you about. In fact, that's not really what I want to tell you about. I want to tell you about some ideas that come through this work, but down into the details, there really are two papers here. One is about cell lines, and the other is about mice, and that's part of the story. We will get there. But the topic really is adoptive T-cell therapy. So for those of you that do not know what it is, you take T cells from the patient, you extract them, you engineer them, and there are different mechanisms of how this can be done. You engineer them to go after the tumor. You introduce them back to the patient and hope that these cells migrate to the tumor after proliferating and then do the job. Seems to work very well in some diseases, in most of them it does not. This is not a talk to understand why it does not work or why it does work or if it's good at all or not, but it is a great example of something that is in the context of cell-based therapy. So it is very important to understand that adoptive T-cell therapy, adoptive therapy, is only one of the mechanisms that fall under the cell-based therapies. And this is a nice slide that actually comes from Pete at the NCI. And you see here a lot of examples. So immunotherapies, including very things that we heard about, like immune checkpoint inhibitors, all these cytokine therapies, dendritic cell therapies, adoptive immunotherapies. That's today, now that is, bone marrow transplant, stem cell therapy. There's a lot of things, and this slide is several years old, so you can imagine that things have kept changing dynamically. So it's a small piece there, but still an important piece. But what is more important is actually this slide. And if nothing else from this talk, if there's nothing else you take home, this is the one interesting one. And that's actually a small story. So that's how I met Pete Schoike, as I said, the head of the brand chief of the molecular imaging lab at the National Cancer Institute. I'm sitting just like you in a talk. Pete stands and gives a talk, and he shows this slide. And in the slide he shows, and the topic of the talk was imaging techniques, so how to image. So they take these, in this case, well, I don't remember if these were CAR T cells or TCR cells. In any event, they take these cells, and they have techniques to image them, how they move over time inside the mouse, in this case, or whatever they put them in. And there are interesting ways of how they do that. In any event, I'm sitting there and I'm looking at this thing and I'm saying, well, you know what? Day 7, day 11, day 14, and day 17, what can they actually do? Is that the best they can do? So at the end of the talk, I raised my hand and I asked Pete, and what frequency can you image things in the mouse? How many time points can you get? He thinks for a second. He looks back at me and says, yeah, probably every 5 minutes, but who will ever want that kind of data? So, well, I would. I do. Okay, so that's how this collaboration started. So this is really, a message. Just communicate. I mean, even on the level of the experimental design, it is super important. I mean, how many times we sit with data and we say, like, what can we do with that? What can we possibly do with that? Why didn't they do whatever? Okay, so that's an example. Anyhow, paper 1 was published in a work on JTB 2020. It is a study of immunotherapy with engineered T cells and IL-2. It is a cell line study, and the experimental setup is not ours. So this is published work in 2018, where they took immunodeficient mice, introduced to 2 cell lines. These are cervical cancer cell lines, a Kasky and 4050. They tried different dosage of TCR-T cells, and on top of it, they played with IL-2. So this is data. And we contacted the authors of that paper, and unlike what happens most of the time, they actually provided us with the data. So we were not involved in any way with that. So that's very interesting. So we had that data and we said, well, let's do something with it. And what we wrote is a model, and the model is not, the details are not particularly important. I mean, you can read the paper if you're really interested, but what you have here are, this is an ODE model with 3 populations. There are cancer cells, the usual stuff, logistic and death as a result of interaction with T cells. The only T cells we are modeling here are, there's a TCR-T cell compartment, and on top of it, there's IL-2, and we allow for, a source term in both of them, the TCR-T cell therapy and the IL-2 therapy. By the way, TCR-T cell therapy, when I say source, it's typically given once. Okay, so just to clarify that. So okay. Anyhow, you do that, and now you have the data. So you can play with the data and calibrate the model. And first, we get the parameters of the tumor growth, because these, for the 2 cell lines, because these are available. Then we add the tumor growth with a TCR T cell treatment. So what you see here is different, the red line is cancer cells, okay? So in this case, it goes up, in this case, it goes down, in this case, it goes down, up, up, and up. You cannot see it's still up. The blue cell is the T cells. We do not have the data for the T cells. The only data that we have are for these dots are the experimental measurements for the cancer load over time. What is different between these 3 columns is the dosage of the TCR therapy that was given at time 0 in the experiment. Okay, so we calibrate the data to that. And then the other thing we can do, I told you there's also an IL-2 in this picture. So why not add that to the model? Well, it was in the model. Let's do something with it. Here's the beauty, okay? Where's the data? Does anyone see the data? Yes, 2 points. Point 1 and point 2. This is the data. What do you think? Right. Actually, 4 points, because there's 2 points for this dosage of TCR T cells and 2 points for this dosage. And what you see here is the IL-2 treatment. There are 3 dosages of that. Okay. Do you want to discuss identifiability of this model? I knew you will do. Okay. Anyhow, regardless, I mean, and there's a whole mathematical analysis behind it, and et cetera, et cetera, et cetera. Well, that's what we have. So that's what we work with, okay? And what do I mean work with? Okay, this is the data. That's all we have. Well, that's not the point we stop. We say, well, what happens if we took the same amount of IL-2 and instead of giving it 3 times, we give it 10 times or whatever, okay, over 3, 4, 5, or 10 days. Well, interestingly, what we observed in our, simulations, of course, you can go back to the lab and conduct the experiments, but it was not our experiment, so, well, they didn't do it, is that there was a difference. You see, this says the 40-50 cell line, and there was the other cell line where he said, regardless of what you're doing with the IL-2, you cannot get rid of cancer in this cell line, and while with the other one, you can. Okay, so that's an observation from simulations. So does that make sense? Well, we did the analysis. I'm just putting here a statement that says, yes, you could actually quantify the difference between the settings where you will be able to get rid of the cancer and not. Yeah, you have to trust the model. But in any event, it does already give a very interesting observation here, clinical. And that is, if this has any meaning to it, well, pay attention to how you determine the dosage of the TCR-T cell therapy. The current practice is based on looking at the patient and providing the therapy based on the weight. That is not enough according to our analysis. You should also take into account the tumor size. Okay, so that's an actual result. Okay, then we move on and we say, okay, oops, how did I, wrong button, sorry. Okay, you finished the talk. No, okay, so the more recent work that was published recently at the Bolton, and it involves mouse models. So what is the other message of a talk like that? We come here and we put a model, and you see 3 equations or 15 or 100 parameters or 2. What we do not show is the amount of work that goes into getting to the point where these models actually work. So of course, if someone gives me now model, an experimental setting, okay, here, there is a mouse, you put you challenge a mouse with a tumor, add engineered T cells, image, okay. I said, like, why do I need a different model than the one that I just showed you on the cell lines? Well, why? Well, the simplest answer is because the other one does not work. Okay, why doesn't it work? Every time we do mouse experiment, you take these different mice and you do something with them, they're identical and you always get different results. So sometimes you get results and they shoot out of the thing and it's not just the math. I mean, actually you see that. So we went on a task to figure out all of a sudden, how can we mathematically explain different results that we get with different mice? And the answer to that was what Peter Lee mentioned yesterday, that's T cell exhaustion. The experiment is you take identical mice, you take an identical dosage of the T cell and you see something different. Well, you can argue that the tumor is different, or you can argue that something is different with the T cells. We took the path of saying something is different with the T cells. Now, we wrote all sorts of papers about T cell exhaustion. That's not a talk on T cell exhaustion, but these are T cells that do all sorts of things. They're certainly less effective in going after the tumor. There's different lineages there. There are all sorts of things we can say about them. Okay, that's separate. What you see here in the model is you see the cancer cells, you see the effector TCR-T cells. We added the regulatory cells, and we added exhausted T cells. If you look carefully at this model, you'll see that the exhausted T cells appear only in the last equation. So why are they there at all? They're there at all because we know how many T cells we introduce at time 0 to the patient. And we speculate, we hypothesize that some of them work and some of them do not. So that's why they're there. But on the analysis level, you can do everything separate than that. And in fact, the analysis is interesting, and you can do that, and you get up to 4 steady states: cancer-free, cancer immune escape, a low tumor that's a dormant state. You can interpret it as that, and a high tumor state. What is interesting here is that a model like that can capture many different types of dynamics. You have cancer. This is a mouse. That's a different mouse. Well, here's another one. There's 6 of them here. Okay. The red curve shows what happens to the cancer. It's the identical experiment. In mouse 1, cancer goes up. In mouse 4, cancer goes down. That's the data. Okay, that's the data about the total immune cell population. Here, by the way, what you see as a separation, the red line is the effector cells. The one above it is the exhausted, and the one below is regulatory. The percentage of exhausted cells is significant. But one model can get all these dynamics. I know there are different parameters, but it's still interesting. If you have that stuff, you can ask what could have been done differently with these mice. Now, remember, the parameters for the mice are different. I do not know how many exhausted cells went into mouse 1 or mouse 2, but you can vary, you can do all the analysis, the settings between these different mice. For example, if you take the number, if you vary the death rates, the death rate that's induced by effector T cell, what happens? Okay, so you increase the death. So basically, these are simulations of different parameters, okay, that this is the data, and the prediction is that we could have done something where cancer would be eliminated if we were only able to play with some values. This is one, that's the other one. Examples here are lowering somehow the number of Tregs. I don't know how to do that, but if I could do that, that would be good. If I could increase the number of effector T cells, that would also be good, and so on and so forth. That being said, I hope I managed to convey the importance of communicating between mathematicians and clinicians / experimentalists. T-cell exhaustion is something that I think we will see more and more picking up in terms of a mechanism that we should consider incorporating into our model. As always, these biological and mathematical systems are much more complex than I pretend they are. And I'd like to thank my sponsors over the years. So thank you for your attention.